Hello and welcome. It's my pleasure to talk to you today briefly about the development of AutoSpill, an improved method for compensating cytometric data. In standard flow cytometry, we deal with spillover of fluorescence into off-target detectors through a process of compensation. Traditionally, the spillover is measured by calculating the difference in signal between a positive and a negative population. This spillover is calculated for each detector, and the inverted matrix is used to compensate, as we see on the right. This is the compensated data. About 5 years ago, we first started using the BD Fax Symphony, setting up panels with over 25 colors. We quickly ran into issues with the compensation. Here's a typical example of what we experienced, showing the uncompensated data here on the left, and the data compensated using single stain cells on the right, using the traditional positive-negative population method. Clearly not great. My immediate solution to this was to go back to the way I had done compensation when I first started, manually adjusting all the coefficients until the controls were correctly compensated. As you might imagine, this takes a while for a 28 by 28 matrix. Moreover, any small errors propagate through the parameters. So I would have to go back and refine the matrix multiple times. To do this, I was using cells staying with the actual markers in our panel, relying on the slope of the data, since most of the markers don't actually bifurcate neatly into positive and negative populations. When Carlos Roca, a mathematician, joined our lab, I sat down with him to discuss how to fix the issues we were having. He realized that the linear nature of quantum mechanics meant the spillover into the off-target detectors would be directly proportional to the signal in the main detector. As such, a linear regression along the slope would be a good strategy. This is the basis of AutoSpill. Moreover, by using robust linear regression, Carlos was able to reduce the distorting influence of autofluorescence on the spillover coefficients. This method gives lower weight to events farther away from the estimated regression line. As you can see here in these examples, this is the standard bi-exponential view, the way we usually look at data. These are the events that are being ignored. And here we're looking at in a linear scale where the straight line of the slope is really obvious. AutoSpill also iterates this process, refining it until the results reach an acceptable threshold. This reduces the error in compensation by an average of 100,000-fold over the standard system. This is particularly important for cases with lower events, less signal, or more autofluorescent noise. What does this actually mean for the user in practice? Well, the workflow is essentially unchanged, and the algorithm is integrated into FlowJo version 10.7 and above now. What changes is that you can use the cells staying with antibodies in your actual panel, and you should get good results. In these examples here, I've tried using traditional compensation or AutoSpill on a panel targeting intracellular cytokines. These are relatively weak signals and rare events that are easily outweighed by the noise of autofluorescence. AutoSpill does a pretty good job. Also, since spread is directly proportional to spillover, the accumulated uncorrected spillover errors in the traditional method are causing spreading. While with AutoSpill, we're instead getting this nicely compressed negative population, actually allowing us a better ability to detect our cytokines. So this project was the brainchild of Carlos Roca. Here, came up with the idea, worked out the math, and created a very useful cytometry analysis script based around it. He's now working as the Associate Director of Bioinformatics at AstraZeneca. Richard Halpert led the team at FlowJo that integrated AutoSpill into FlowJo. And this work would not have been possible without the support of the ERC, VIB, and KUL, or without the guidance of Adrian Liston. Thanks for listening.